I wanted to start off on a little bit of a different note. About a week ago, I was in our offices in my clinic, and Dan Peterson had just been visiting the day before, and I was mulling over some of the pearls of wisdom he left me, and the usual chaos was erupting around the clinic. We had a couple of Rituxan patients in, a new patient was visiting, and litany of research issues waiting for my attention. And the phone rang, and everything kind of came screeching to a halt. And it was one of those calls that no clinician wants to get, no patient, no family member wants to make. And we had lost a patient. A 17-year-old young man had been sick for about 2 years. This is him prior to being ill. And it really struck us hard because this was a failure of our system. And here we were in a place where we were seeing so much promise. I had seen him maybe a month prior to that, and really he hadn't embarked on any kind of a treatment path yet. But it just made us really realize that for the patients out there, you all don't always have as clear a view of what's going on. And so I think if there's one takeaway from my talk, and hopefully from the whole day that you've gotten today, is that really there is a huge sense of optimism out there. And the field itself is in a place, I think, in a better place than it's been in its entire existence, for my opinion. So if there's one point I get across today, it's that, optimism. I think there's so much going on, and you've heard about some of those things, and I'll try and wrap some of those things together with what we're doing and where we hope the field is going in the next 12 months. So we've heard about treatments that are beginning to emerge, obviously the Rituxan treatments being the big splash, and I think that's been, as Jorgen put it the other night, a game changer. We've seen a lot of mainstream science getting interested, a lot of scientists who would otherwise not have been interested in chronic fatigue, and we're, I think, all trying to accelerate that process. And we've seen some of that at the meeting here and the meetings for the last couple of days. And I think that's a critical piece to moving this forward, actually, and I'll talk more about that. We've seen uses of different technologies to advance our understanding, and whether it's measuring various immunologic parameters to information technologies to the latest pharmaceuticals, but I think we need to be very smart and strategic about that, but we're making headway on these. And more importantly, I think as a field, we're approaching a critical mass. I think there's a lot of collaboration. You just heard a long list of projects from Dr. Peterson around how we can work together, and indeed many of us are working together, and I think that holds enormous promise. I wanted to just start on that path with that thought. Some of you may not be familiar with us, so I'd start with 2 words about that and the direction we've started out. I used to be at Stanford, and I left about 3 years ago to start OMI, Open Medicine Institute. And really one of the founding principles was that clinical medicine and research have really been driven apart. I mean, I think we're extremely fortunate in this field that we have some very astute and curious and strategic physicians like Dan Peterson, like Olav and Oysten, who saw things in their routine clinical practices that they then brought out and have pursued quite persistently. And we all have that to stand on and build on. But the typical clinical practice, particularly in the United States, but also here in Europe and elsewhere, has been divided from research and from physicians asking questions, because the visits get shorter and shorter, and the time available to physicians is less and less to actually treat their patients. And that's a huge problem. It's a systemic problem that we have, and I think it becomes particularly acute for chronic fatigue syndrome, ME. So we started the Institute with the idea of bringing those 2 disciplines essentially back together. And there's been a huge push, lately for translational medicine, but we wanted to do it in a way that brought it together particularly acutely using biotechnology, using information technology as a basis for this, and leveraging social networking, and I'll talk about exactly what I mean by that, and biosampling, and I'll get into each of those. So last year I threw this slide at you as well and said one of the problems in chronic fatigue syndrome and why we haven't moved particularly far is that syndromic diseases are difficult. They're difficult from a lot of perspectives. They're difficult because they're these mixed bag collections of symptoms. The diseases definitions, as you know, aren't mature. Biomarkers are scarce, and we're doing lots of things in lots of corners that may or may not have effects, and that's hard to measure, and there's not a whole lot of standardization, and outcomes aren't really followed clearly. So I wanted to take a survey before I dive in of how disease is accepted today. How many people, just quick show of hands, think peptic ulcer disease is how many people think that the general populace thinks peptic ulcer disease is a real disease? Hopefully everybody. Okay, well, not everybody, but Parkinson's disease. Okay, most of you. Alzheimer's disease. Okay. Multiple sclerosis. Most of you. MECFS. This is a biased audience. So, same question. Roll back 20 years. Okay, so peptic ulcer disease. How many people think that 20 years ago peptic ulcer disease was taken seriously by the average by the general populace? So about less than half, I'd say. Parkinson's disease. A few more. Okay, maybe half. Alzheimer's. About the same. MS. A little less. MECFS. Nobody. All right. Well, if you look at each one of these syndromes, each one has gone through an evolution that actually has very much paralleled where we are today with CFS. Peptic ulcer disease was actually probably diagnosed or first thought about prior to 1958, but it was only in 1958 where the first association to a bacteria actually causing an ulcer in the gut was made. And it wasn't really until 1997 when the CDC finally approved triple antibiotic therapy, and it was only 2003, somebody correct me, when Marshall and blanking on the other guy's name got the Nobel Prize for this. So this has been a disease that people did not take seriously. Marshall was laughed out of the room in Australia when he first proposed these things, and it took essentially a generation of physicians to change that attitude. Parkinson's has been going on for a lot longer, obviously, but also along with Alzheimer's, which was also mentioned today earlier, hasn't really come into its own until the 70s and 80s. And MS even later than that, which has been the first diagnosis of MS was in 1868. So these are syndromic diseases that have very slowly evolved and slowly been accepted. And MECFS, in that respect, is actually quite young. And I would be the last person to suggest that we should wait for any length of these kinds of timelines, because I think we're now in a different era where we can actually do a lot more. But I also caution you that we have a lot more to do and a lot more attitudes to change, which I think I don't need to labor that in this audience in particular. So let me just spend a few more minutes on that concept and dig into the peptic ulcer disease story. So here's a disease that went from a syndrome to a treatable disease in 1997. And now, while I was at Stanford, I worked in a group where we actually looked at H. pylori in much more detail. And at the genomic level, we could go in and take and biopsy an ulcer site and normal tissue next to it. And the normal tissue would give us a gene pattern, a gene expression pattern. I've summarized it here with 9 different colors, because we like colors on our little microarrays. And then I can biopsy the ulcer itself, and I get a different pattern, a very different pattern, a very distinct pattern saying that there is pathology there. And I could look at the path slide and do that as well. But as I look at the path slides going through there, if I continually biopsy, I'm going to see more subtle differences. Whereas if I go and look at the gene expression, I'm going to see actually much more definable, much more rigorous changes here. So the infection is going to progress to a precancerous state, and if it's not treated, that would branch off to a fully cancerous state. But if it's treated, it's going to start to regress, and it's going to go back towards a resolved type of profile, which may not perfectly match the normal, but it's pretty close. But that gives us a way to diagnostically and extremely definitively say this patient was normal, and sorry, I don't have a pointer, but I'll use the mouse. This patient was normal, or we put the patient on treatment and we put them on the wrong treatment so they're not moving, or we put them on the right treatment and they are actually resolving back towards normal. And it's a principle that I think we need to apply in CFS. So we've done actually a lot of that in the last year, and in the last few years even, and we're starting to get some of those things coming out. I think Jonathan Kurz was here in the UK, was one of the first shots off the bow when he clustered CFS patients into a few distinct groups, 8 distinct groups, that had different expression profiles. And it was a very limited gene expression analysis, but it was particularly useful, I thought, nonetheless. And it really started to set the stage for where we are now in saying there is the potential to define this disease in other ways other than as a syndrome or collection of symptoms. More recently, there's been a proteomic separation of chronic fatigue syndrome and Lyme disease through spinal fluid. So looking at proteins in the spinal fluid, we can tell the difference, or at least we appear to be able to tell the difference. These are still very small studies between CFS and Lyme disease in the spinal fluid. Huge issue, right? We've always thought about overlap, and obviously that needs to be validated and so forth. Likewise, there appears to be a limited gene signal allowing us to differentiate major depression from at least fibromyalgia, possibly crossing over into chronic fatigue, but that's huge too. Just scoop out the depressed patients, right, and say, okay, depression is not the issue here. So that, obviously, I'm sure many of you have heard from physicians, oh, you're depressed, or oh, this is psychiatric. I know here in the UK is a particularly biting issue. So this would allow us, if we can develop these things, to really take these molecular things and apply them directly into a clinical problem, right? To tell that GP, re-educate us to what really is going on. Okay, so the last 12 months have been particularly exciting. And you've heard about the rituximab trial, which was, as I said, a game changer. The XMRV work, so to date, has been debunked, and Dan Peterson just told you about hopefully the final analysis of the final study on that. Shortly after we did that work with a wonderful collaborative group that we pulled together with Dan's help, we got word of a little bit of a change in the winds, and the CDC was looking to actually go back and try to recharacterize, and there was, I think, a shuffling of the guard there as well, which may have enabled some of this. But for the first time in a long time, the CDC was reaching out to try to do something, I think, somewhat innovative and at least something that we saw that could be useful in the field. And so that in and of itself, to me, is a landmark event. Right here we have a government agency potentially turning the corner on its attitude towards CFS. We've continued some of the rituximab experience in coordination with the Norwegian group, and that's been very interesting, and I think really has started to put a positive second site spin on the data that you've already heard about, but that's ongoing, and I'll talk about that in a little bit. An Ampligen study was published finally, which was not the biggest splash, but at least there was some positive notes there to hang on, and so we had yet another treatment route that has having a real effect. And I already mentioned the other 2 studies in terms of diagnostic. So where are we? What are we doing at OMI? And I'll talk in a minute about how we're doing it. I'm going to kind of just gloss over this in the interest of time because there's too many things that I'd like to cover. We've actually been taking a group approach to to this. So we've spent much of the last 2 years building a consensus, building a network of physicians, a network of researchers, a network of any interested party, be it a pharma, be it a diagnostic company, a government agency, whomever that wants to do work in the area, and tying all of those together so that we have coordinated standards. Some of the things that Dan Peterson had up on his slides, we have coordinated standards for laboratory sample collection, that we have really good and rigorous standards for how those samples are processed, how any kind of data is collected, and how we approach the different treatment regimens, and making sure that we collect across multiple sites and so forth, really good outcomes data on anything that we do. So with that in mind, we've embarked around investigations around looking at various things that have shown potential as treatments and shown potential as diagnostics, and kind of looking for some of the low-hanging fruit that we haven't really analyzed fully. So we have actually an ongoing a study looking at viral flora in the oropharynx, so in particular at herpes viruses, so quantifying those, and that's starting to yield some very interesting data. We've been doing autoantibody arrays, sort of pan array looking similar to what the Norwegians ran, but probably the next generation of that. One of the advantages that we have where we are, Mountain View, is really right smack in the middle of Silicon Valley. And Silicon Valley is these days actually quite misnamed. It's actually much more of a biotech valley. There's more pharma startups and more diagnostic startups there than I can count, and certainly more than a lot of the IT startups. But there's also a lot of these social networking, so there still is that flare of information technology sort of permeating, and we can pretty much walk down the street and find a lot of these resources, which is precisely what we did in setting up a lot of these collaborations. So the viral flora study, for example, is with a company that is trying to build a herpes viral diagnostic. And so we were the perfect platform for them to try it out and validate it for them, or debunk it one way or another. We were also doing cytokine arrays, which were developed right up the street at Stanford, and in collaboration with our good friends at the Stanford Genome Technology Center, we're doing some deep sequencing, as well as with another startup in San Francisco that's doing deep sequencing for free, actually, on all of our clinical patients, on all our clinical samples. They'll give us the sequence back. So we should have some interesting data coming in the next year or so on that. And that's intron and exon regions. Ongoing treatment studies. Some of these are pilots, some of these are more in-depth, and I'm not going to dive into a lot of depth. You're welcome to ask questions afterwards, or if you'd like. Valganciclovir, I'll give you a quick update on that. The EVOLVE study that Jose Montoya and I ran at Stanford has finally gone to publication, and I don't think it's been accepted yet for publication, but we'll see where that goes, but hopefully that should be out soon. We've continued with several other pilots around valganciclovir, which are showing actually quite positive data, which is exciting. But also is not as positive as our Rituxan patients in terms of the rates of response. We've actually done a number of pilots with several other antivirals already prior to Rituxan, and there's several of those that have actually seen some very positive responses as well. Anti-inflammatories haven't been as interesting, but those are ongoing. We are doing a follow-on, essentially, to the work done in Norway, with rituximab, off-label, essentially pilot study, simply because we haven't gotten the funding yet for doing a multi-center trial internationally, which is what we hope in coordination with the Norwegians to get off the ground. And we've looked various places for funding, and I'll talk about that in just a little bit. Rifaximin is another drug. It's an antibiotic, actually, that has shown promise in a much more limited subset of patients, but in some patients it literally gets them out of bed and completely functional again. You know, we see relapses as we've seen with Rituxan or Valcide or anything else with this drug, and it opens up the question, you know, which pathways are these are any of these treatments hitting? And Øystein already hinted at that there may be common pathways here, there may not be common pathways here. We actually have no idea, and we're looking at those very carefully, but I think that's one of the low-hanging fruits that I'll talk about later. And then, we're certainly open to doing other non-pharma interventions. We've had a study that hasn't really gotten off the ground too far yet because there's some device issues with it, but we had talked about regional and actually whole body hyperthermia as a potential intervention, which had shown some early promise, but there's some technical issues with doing that in terms of the device. So let me shift gears a moment with that in mind and to the comments around whether, is CFS ME autoimmune? Is it viral? Is it bacterial? And we've had these questions kind of circulating around the room. And I would put to you that we don't know. And we need to look at that. It may be all of the above. It may be some combination. One could easily postulate an NK cell deficit leading to herpes viral or other intracellular infection that, there's certainly an NK B cell cross-connect in terms of signaling, and so if you eliminate the B cells, is that somehow resetting that signal process? You could also look at herpes viruses infecting the B cells. We know that happens. We know B cells traffic some of these viruses into the CNS, so are those involved? And so some of our studies have been aimed at that. Some of our Rituxan patients are actually on antivirals, so it's combination therapy, and we'll see where that goes. But certainly those pilots will not be the end-all, be-all. We need larger studies, as Øystein and Olaf suggested. We need much larger studies to really validate any and all of these potential origins. But I just put up here a lot of the derangements that we've seen, and some of these we see improving when patients improve. And some patients, simple dietary changes have made all the difference. Other patients, you can change all the dietary issues you want and you will get nowhere. And we need to sort of start differentiating and putting patients into different tracks, so to speak. So that's a lot about what we're trying to do. And the way we're doing that has been to build out a network, and build out a network of collaborators, build out a network of clinicians, build out a network of patients, and really focus on how do we connect patients, how do we gather data directly from you all, how do we gather data from each other, from physicians, and put it into a common platform or a common database or some way that it can be shared even if it's not in the common database. How do we interact, and how do we get pharma, whether that's the pills down in the corner, or diagnostic companies to interact with us more? And how do we, these are patients down here, how do we interact with patients directly? And we've gone about that in a number of ways, quite strategically. And you'll see some things coming out at us, and you'll sort of scratch your head and probably wonder, well, are they really interested in CFS? But we are, I can assure you of that. And the reason why you'll see some things coming out that may have much more generic labels at it is because actually some of these things are much more applicable to all of medicine. We've forgotten how to do research in a lot of medicine. And part of our agenda is to take not only science back into medicine, but take science back into CFS and really make it a much more mainstream process, which I think hopefully ultimately will help gain larger acceptance for the disease as well as for the work that needs to be done in the disease. So how are we doing that? Well, thankfully we have a lot of willing collaborators, many of which are around the room, and I'll go over some of that later. But really the core of what we're doing is something we're calling OpenMedNet, which will go live later this month. It's already live, but it's live internally. We'll turn it on for the general public, and I invite all of you to come now. I'll have the website at the end. But OpenMedNet really is an informatics platform that allows us to communicate, and it allows us to communicate in a lot of different ways. Users, patients, providers, researchers can come in over the web. They can come in via smartphone or iPad. They can come in sometimes via regular telephone. There's always back-end data feeds into hospital systems and lab systems, as Dan was talking about. We have two-way interfaces with faxes. Well, how do you do a two-way interface with a fax? Well, you send someone a fax that has a bunch of checkboxes and things. It's a form. They fill it out, and they fax it back. And it has object character recognition technology that can see what they filled out and can interpret some of those things and put those directly back into a database. It can send patients and providers email or text messages, and you can have interactive sessions to check in on a patient, right? How are you doing today? We don't do that as physicians. I see a patient, and I'm lucky if they come back to me in 2, 4, 6, 8, 10 weeks. I have very little connectivity to them, and then I have to ask them all sorts of questions when they come back. And in the meantime, I'm in my office wondering, what's going on with them? Are they doing okay? Or I get a cryptic message, and then I have to call them, which I love doing. But it's much better if we can be preemptive about this rather than and proactive and preventive rather than reactive all the time. So if we're measuring and monitoring all the time, we're using various different home monitoring devices, which are also hooked into OpenMedNet, and the Norwegians also talked about one of those, the SenseWear, which is an armband that many of us have patients wearing, but there's many other devices that can be hooked into this. So I kind of probably have talked about everything on this slide already, but really the key here is better connectivity, better communication between the patient, the physician, and in this case, the researcher as well. So where are we today? Well, we have 12 plus clinical sites. We have 3 national clinical labs participating, 3 major hospitals. We have a large biorepository with over 10,000 patients in it already. We have a data repository, that's what that's supposed to be, with an IT infrastructure that has that's actually approaching, I think it's actually closing on 100,000 patients. These are not all CFS patients, I'm talking patients in general. There's 10 plus academic sites, 12 plus research labs. Lots of patient groups have gotten interested, and we're on 4 continents, so no Africa or South America or Antarctica yet. And part of the way we wanted to get this more into the mainstream was we looked around and found some really key innovators. I guess all of them are on the West Coast now that I think about it, but that have helped us formulate this. And actually many of them are interested in CFS and engaged in CFS. Obviously Dan Peterson, you all know. So he's our representative clinician on this panel. Ron Davis is one of the dreamers of the Human Genome Project and actually one of the people who made sure it successfully was implemented. And he is extremely engaged in CFS and happens to be my neighbor. And I've worked with him for about 5 years at Stanford. Tom Fogarty is a name that may not be so familiar in this field, but some of you may have heard of him. He's a brilliant innovator and is actually the inventor of the cardiac cath, which is the basis for all cardiac catheterization in the world today. Also left Stanford and happens to have an office on the floor above me, or in my old office. So has been a really huge support. Randy Scott is a PhD chemist turned biologist who has successfully started a number of different genomic and genetic companies in the U.S., the latest of which is Genomic Health, which is a company that has the most successful, certainly, and I think possibly the only commercial genomic panel array for a test called Oncotype DX, and they've got several others that they're working on now, but this is a test that can be done on a breast cancer specimen that tells the clinician, and I don't know, our oncologist here may want to comment on it more than I can, but that tells the clinician in certain subsets of patients whether that patient should get chemotherapy or not. So it can potentially save a decision for a very expensive therapy, but also one that has a lot of morbidity. And there's all kinds of interesting stories around that in terms of how that's been applied and how it's been accepted or not by oncologists. And I'd actually be interested in the European experience on that. And Jay Levy is one of the discoverers of HIV and has been a leading researcher at UCSF on the immunology and virology front for many years. So we use these folks to kind of guide us, and I'll have a little thing at the end, a video from some comments from Randy Scott, just to kind of whet your appetite. Okay, yeah. So just lots of folks participating. You know lots of these names. I'm going to keep moving right along. Lots of different institutions, and the key is really how do we wrap them together. Many of these institutions you know and love. So this is Randy's little video here. I think it's a minute and a half long.
The model for open medicine really is the model for the future. How do you connect the patient and their genomic information? You need the patient, you need the sample, you need quality genomic information, molecular data about their sample, but you need to link that into their treating physician who really knows a lot about the phenotype of the disease and the pathology features of the disease. Those aren't going away. Those are critically important. So the question is not that will genetics transform medicine by itself. It's going to be when you bring genetics in and meet up with the traditional clinical and pathology information, and now you can begin to link that medical treating physician with the broader research community and say, where are there other individuals that share some of these same features, some of the same clinical phenotypes, but also some of the same genetic information? How do we build this into a broad, sort of open network of shared information that really transforms how we treat and think about disease?
So we'll have a longer video on that on the website in a couple of weeks. So let me just say a few words about crowdsourcing, and then I'll wrap it up. So really a big push of that, as Randy indicated and I indicated, was getting people involved and getting data from people. So in terms of crowdsourcing, we need data from lots and lots of people. We need that to be able to do some of these pilots, and we need it to be able to actually successfully execute clinical trials on the right subgroups of patients and so forth. So we're getting data from various collaborators, and we're having a meeting in June in New York to gather folks together for CFS in particular, the merit meeting, to set practical targets around what is our agenda, what do we need to hit in the next year, who is going to do it, and how are we going to fund it. And so we'll have some news coming out after that meeting in terms of what gets set at that meeting, and how do we work together to do it. We will look to do a roughly a $5 to $10 million fundraise, and we have some donors identified to do that as well already. But we're also looking to crowdsource that. So if we can find 50,000 people who are willing to donate 100,000 each, well, then we have a Rituxan study funded, right? And we want you all to weigh in and say, well, we really want a Rituxan study, but okay, that's going to cost $5 million for a 100-patient Rituxan study, or whatever the number is, drug alone, I think. Or should we go after the smaller study, that $100,000 gene array study that we think might yield something? So we're going to put some of those projects out on the web after the merit meeting and say, here's our priority list, here's what we think it's going to cost, and here's how much money we have to date to do it. And here's where we can go, and here's where we're going to look for more money, or come help us find more money to do that. And here's the people involved in doing it. So really, again, bringing rigorous science and people together with a collaborative group to get these things done. So I talked about all these things, I think. So how can you help? Send us your information. When the website goes live, log on, enter your own information. If you're interested, we can send you a kit, and you can sample, or you can get sampled at one of our sites. That's blood, CSF, anything. We're looking at lots of other samples, bone marrow and so forth, and when someone's talked about brains as well. Contribute to the network. And tell your friends and neighbors, because one of the things we're going to try and do is put resources out there that people can use. So I'm pretty much done. So hopefully June 18th, you'll see some changes on our website around the network and information, and then the OpenMedNet, which is where you can log in, and there'll be some resources to help you all manage things there as well. So thank you for your attention, and I think, again, really the take-home message is that there's an incredible amount going on, and you should all be very optimistic, because we are. So thank you.